my name is Rebecca. Is that volume too loud for me? My name is Rebecca. I'm a third-year graduate student in Harvard's Biological and Biomedical Sciences PhD program. And today I'm going to be talking to you about genetics and genomics and obesity. Okay, so I am now showing you a list. You can take a couple seconds to look at it. And if I asked you what all of these things have in common, you'd probably say these are all human traits. Some of them are human diseases, but they're all sort of attributes that humans have, and you'd be right. But the more specific answer that I am looking for is that all of these things are influenced by genetics. So all of these things have at least some genetic component to them. And even more specifically, all of these things have been studied in the genomics revolution, which is just using big genetic data to uncover the causes of human traits and diseases. So using data collected across thousands of people or tens of thousands of people or even hundreds of thousands of people to study these traits and what causes them. So today I'm going to be focusing on obesity as my particular trait of interest, but I just want to take this second to remind you that the principles that I'm going to be talking about in this lecture are really applicable more broadly to the study of all of these different things. And so this is actually sort of a foundational topic for the study of lots of cool things that are happening today. So just to give you sort of an outline of what I'm going to cover, we're first going to talk about obesity risk and some basics of human genetics. In the second section, we're going to talk about monogenic obesity, which is obesity primarily caused by one gene. And don't worry, we're going to talk a lot about what that means before we get there. And then finally, we'll talk about polygenic obesity, which is obesity that's caused by multiple genes. Okay, so let's start with some basics. And you might wonder, sort of, how do we talk about obesity in a scientific way, because it's sort of a qualitative trait. So the measure that scientists often use is called BMI, which stands for body mass index. This is a term you may have come across in a doctor's office. It's just calculated as weight divided by height squared in kilograms and meters. It's a relatively common measure of obesity, and a BMI of greater than about 30 is usually the clinical definition of obese. So as most of you are probably aware, obesity is a worldwide epidemic, and particularly in America, an epidemic. So between 1980 and 2008, the worldwide prevalence of obesity is almost doubled. And if you look at this graph here, the y-axis or the x-axis shows years from 1999 to 2014, and the y-axis shows the percentage of people who qualify for the clinical definition of obesity in that year. So the blue line represents adults, and the green line represents youth. And you can see that in 1999, about 30% of adults qualified as obese, whereas in 2014, almost 38% of adults qualified as obese. So that means more than 1/3 of adult Americans currently are considered obese, which is a sort of astonishing level of prevalence. And the reason, of course, that we care about obesity from a health point of view is not so much that being obese that's an issue as the health consequences that tend to come with it. So some of the main ones are heart disease, diabetes, some cancers, osteoarthritis, which is a disease of joints and joint pain, high blood pressure, and breathing problems. So given the fact that obesity has such a high prevalence, both in America and in the world more generally, and that it's associated with all of these negative health outcomes, I think you'll all agree with me that this is a problem that's worth devoting some of our time and research hours. Okay, so what actually puts a person at risk of becoming obese? So if I drew a scale, and on the left I said not obese, and on the right I said obese, how do we get the scale to tip in favor of obesity? What goes into that? So one thing is a poor diet. So if you eat a poor diet, you're going to be more likely to be obese. If you don't exercise, if you lead a sedentary lifestyle, you're also more likely to be obese. And there are medications that you can take which have weight gain as a side effect. So those would also increase your risk of being obese. And finally, there's DNA or genetics, which is obviously what I'm going to be spending most of this talk discussing, but just to remind you, this is really all in context. So there are a lot of lifestyle things that go into obesity risk beyond just genetics. Okay, so let's get us on the same page here. So some of you guys may be familiar with DNA to varying degrees. But DNA is essentially the blueprint of life. It's the instructions for making all of the parts of you. So DNA contains 4 sort of molecular units, which we call bases. So the bases are adenine, guanine, cytosine, and thymine. And we usually just abbreviate them as A, G, C, and T. So that's what I'll be doing for the rest of this lecture. And the sequence of these bases, so the order that these bases take in the DNA, is the instructions that make up you. So what does that look like? Well, if we zoomed in on one section of DNA, it might look something like this. So you see this sort of long sequence of A's and T's and C's and G's, kind of just all in what is apparently, although it isn't, a random order. And it turns out that's all your instructions, which I think is pretty cool. So a complete set of DNA, so all of the instructions. Whoa. That's cool. So a complete set of DNA, so something that requires that all of the instructions to make all the different parts of you, is one genome. So in humans, one genome is about 3 billion bases. So if we go back to my illustration of a DNA sequence, imagine 3 billion of those letters. So it's a pretty unfathomable scale, in my opinion. So that's really when we say genomics, that's what we're talking about. Genomics is just the study of genomes. So it's looking at all 3 billion of those bases, which you can imagine is a pretty complex problem. Okay, so if we take a person who happens to be in genome, and we look at a particular part of her DNA. Let's say in that part of her DNA, she has ATCA-TTG. Okay, so now we have another person named Amy, and we're going to look in the exact same spot in her DNA. And she has almost the same DNA sequence, but where Tina has a T, Amy has an A. So you can see the bolded red T in Amy is a bolded red A in Amy. So that's a single letter difference. So we actually have a name for that difference. We call it a SNP. In this case, this would be a T to A SNP. So SNP stands for single nucleotide polymorphism. Nucleotide is just another word for base. Polymorphism is a word that sort of just means difference or change. And all it is a location where a base in the DNA can be different between 2 people. So like we just talked about, where Tina has a T, Amy has an A. So that spot is a SNP because not everyone is the same. Now these actually exist all over the genome. So I told you there are 3 billion bases. There are actually millions of locations in the genome where it turns out that people can be different from each other. So even though we are much more alike than different in terms of DNA, we're something like 99.5 to 99.9% alike, that's actually still a lot of places where the DNA can be different, right? It's 3 billion bases, that's a lot of locations. So this is actually part of what makes us different from each other, is that we all have a different set of SNPs, unless you have an identical twin. So where do SNPs come from? So everyone inherits 2 copies of each SNP, and that's because you inherit a full copy of the genome from your mother and another full copy of the genome from your father, and those combined make up your DNA. So as a result of that, you're inheriting 2 copies of each SNP. You're inheriting one from your mother, and you're inheriting one from your father. So to take an example, if we take this random SNP here, so this is a T to G SNP, there are 3 possibilities for what a given person could be. They could be TT, so they inherited a T from their mother and a T from their father. Or they could be TG, so they inherited a T from one of their parents and a G from the other parent. Or they can be GG, so they inherited a G from their mother and a G from their father. So for this particular SNP, this location in the genome, every person in the world is going to be in one of those 3 populations. Okay, so going back to obesity, so how much does DNA actually tip the obesity scale? And by that, I mean, does it kind of look like what we see on the left, on the right? Is DNA contributing a tiny amount of risk for obesity, or is it contributing a huge amount of risk for obesity? And the answer is actually that it depends. So the reason that I just spent a bunch of time talking to you about SNPs is that not all SNPs are created equal. So we're actually going to be talking about kind of two different categories of SNP with respect to their effect on risk. So what we see on the left is what we call a SNP of small effect size. So that's a SNP that's only going to increase your risk of obesity by a teeny tiny bit. By way of analogy, if we go back to this idea of a TA SNP that we have in this ATC sequence that I have up here, you can think of that as if I took the word gray, spelled G-R-A-Y, and I changed the letter A to the letter E. So if we change the spelling of gray like that, so G-R-A-Y to G-R-E-Y, even though we've changed one letter, the meaning of the word hasn't really changed very much at all, right? So it's the same concept. We've got a single letter change, but from the point of view of the meaning, it hasn't really made that much of a difference. However, you can have SNPs of large effect size. So let's say we have a G to C SNP now. And imagine taking the word gray, and this time, instead of changing the A to an E, we're going to change the G to a T. So G-R-A-Y becomes T-R-A-Y. So now we've really changed the meaning of the word, right? Because the word gray and the word tray really don't have that much in common with each other in terms of what they actually mean. And so a large effect SNP is like that. So even though, just like the first example, it's a single letter change, it actually changes the instructions so much that you get a really different result, and that causes a big change and therefore a large effect. So all of this together is to say that there are actually two kinds of obesity with respect to genetics. So obesity can actually be monogenic, which means it's caused mainly by one very large effect SNP, or it can be polygenic, which means it's actually caused by small risk from many different SNPs, as well as large changes that have to do with lifestyle. So from a pictorial point of view, if we look at what's on the left, that would be monogenic. So a massive contribution from DNA with a relatively much smaller contribution from lifestyle. Whereas polygenic is like what you see on the right, where there's huge contributions from lifestyle, and then a bunch of contributions from various parts of your DNA, but all of them are very, very small. Okay, and just to put a number on this, I'm going to tell you to take this with a massive grain of salt because it's extremely difficult to estimate these numbers correctly. But I would say that something like less than 5% of severe and early onset obesity would qualify as monogenic. So it's very, very rare to have monogenic obesity. The vast majority of the time, obesity is polygenic. Okay, so I just told you that polygenic obesity is much more common than monogenic obesity. But why do you think that is? So if we go back to Tina and Amy, and we had this sort of T and we had an A, and we said that that's a T to A SNP, I'm going to give you an additional piece of information. So the additional piece of information is that we're now going to step back and look at everyone in the world and say the percentage of people who have a T in this spot is 60%, and the percentage of people who have an A in this spot is 40%. So that's sort of another dimension of this SNP, and if we look at it in a different way, I think that might be more clear. So if you think of 100 people, so that's 100 blue dots, each of those is a person, 60% of people carry a C, and 40% of people carry an A. So 60% of them are green C carriers, and 40% of them are A purple carriers. So if I saw this SNP, I would say this SNP is common. Because if we look at either version of the SNP, 60%, 40%, a lot of people are walking around with both of those versions. But if we took 100 people, and now 97% of people carry a C, and only 3% of people carry an A, I would say that that SNP is rare. Because if I pull the person at random from the population, the chances that I would pull someone who had an A at that location would be really, really small, right? Most people are walking around with a C. And it turns out that this distinction between common SNPs and rare SNPs is actually very important. So in general, this is just a heuristic, but in general, we call a SNP common if both versions of the SNP are present in more than 5% of people. And we call it rare if one of the versions of the SNP is present in less than 5% of people. Okay, so we just talked about two important properties of SNPs. We talked about their effect, so how much they increase your risk of obesity, and we talked about their frequency in the population. So the question that I'm sure is now burning in all of your minds is, how are these two quantities related to each other? And the answer is that they are related to each other. And the way that they are related to each other is that large effect SNPs tend to be rare, and common SNPs tend to have small effects. So why is that? The reason is natural selection. So some of you may be more familiar with the concept of natural selection than others, but essentially the idea here is that if you have a SNP that has a strong negative effect on your health, you are going to be very unlikely, or less likely at least than someone who doesn't have that SNP, to reproduce and pass that SNP on to the next generation. And so if a SNP exists that causes a really, really bad health effect, it's not going to make it to being common because the people who have it are not going to have children as much as the people who don't have it. And so that actually turns out to have a pretty pronounced effect on the distribution of different versions of SNPs that we see in the population. So if we look along an axis of a SNP being rare to common, so along the x-axis, and then on the y-axis we have the size of the SNP effect. So a small increase in risk at the bottom, a big increase in risk at the top. There are a bunch of categories that SNP could fall into. So most monogenic obesity SNPs are going to fall into this category of being rare with a big increase in risk. In contrast, when we talk about polygenic obesity SNPs, these are going to be common with a small increase in risk. And if we wanted to know about common SNPs that cause a big increase in risk, you would discover that there are actually very few SNPs in that category. And all of this is for the same reason of natural selection, that you're not going to get a lot of SNPs that are both common and cause a big increase in risk. Because if you're a SNP that causes a big increase in risk, you're probably going to make it less likely that the person you're in is going to have children and pass it on. And finally, you may be wondering about this last quadrant, which would be rare SNPs that cause a small increase in risk. And in fact, there probably are a lot of these that contribute to polygenic obesity, but it turns out these are statistically kind of a nightmare, and we don't just have the tools to deal with them right now. So they're very interesting, but for right now, we're just going to call that biblical. So, and just to take a step back for a second, this breakdown of monogenic versus polygenic SNPs and the sort of patterns that they show for obesity is actually true for a lot of other traits. So it turns out pretty much most of the traits you can think of are probably polygenic, but some of them, like obesity, actually also have monogenic versions where there's some SNP out there that has a really, really strong effect and is probably also really, really rare that can kind of cause the trait by itself. So other examples are height, Alzheimer's disease, diabetes, cardiovascular disease, epilepsy. All of these are generally polygenic, but actually have monogenic versions, which I think is pretty interesting. So in summary, obesity risk is a mixture of lifestyle and genetics, and we talked about two important properties of SNPs. So we talked about their effect, so how much they increase your risk of obesity, and their frequency in the population. In the next section, we're going to talk about monogenic obesity, so rare and large effect SNPs. And in the third section, we're going to talk about polygenic obesity, so common and small effect SNPs. And I will now take any questions you have.
Yeah, when it comes to obesity, there's one difference I think that for our ancestors, the ability to store fat was vital to your survival. So it wasn't a bad trait to have. And our life, but we no longer have famines, and but we haven't had enough time for our bodies to adjust to that. So does that have something to affect that?
Yes, so the question is, wouldn't actually there have been, would negative selection have worked the way that I just said it works for obesity, because perhaps in the past, the ability to store fat would have actually been selected for positively as opposed to negatively. So that's actually a really good question, and that's something that scientists have thought about for quite some time. So what you're referring to is actually called the thrifty gene hypothesis, which suggests that humans have been selected for genes that are going to actually make you store fat and gain weight, because in the absence of food, that wouldn't have been such a problem. There is not a ton of evidence to suggest that's true, although it's an appealing theory. But I think the bigger issue, actually, which is sort of a subtle point, is that most rare variants that cause, rare SNPs that cause large effects, usually carry other negative health consequences. So if you have, we're going to talk about monogenic obesity in just a second, but 2 out of the 3 examples I'm going to show you also come with something called hypogonadism, which is issues with development of the gonads, so the ovaries and the testes, which you can imagine would lead to pretty strong negative effects on your ability to reproduce. So obesity is sort of a weirdly subtle example for this, but I think it's actually fair to say that even if selection isn't necessarily acting on the specific trait that you're looking at, so in this case that would be obesity, having a SNP that has a huge effect on something is probably going to mess up something else in a way that is pretty detrimental to your health.
Does epigenetic have an effect on SNPs, or?
So the question is, does epigenetics have an effect on SNPs? So that's a good question. So certainly, I think that there are SNPs where the effect of the SNP is to change epigenetic marks in the genome. So epigenetics, if I don't know if all of you are familiar, is this idea that you can sort of put things on top of DNA, so you can like methylate it, which makes it, silences it in a way, or you can sort of up and down sort of how the instructions are being read. So I think you can generally think of we think of SNPs and epigenetics usually separately as like different things that can affect a trait, but they can be related, like I just said, that like you can have SNPs that will change the epigenetic profile of DNA. So that would be the mechanism by which a SNP is exerting an effect, if that makes sense.
I got 2 questions, one was quick. First is, are SNPs always binary, or are they sometimes like 3 or 4?
Good questions today, guys.
And second question is kind of related to hers. Is the data indicate at all whether in societies that have like been able to produce more food, better health care, if any of those rare SNP combinations have increased in frequency? You talk about how they were scaled down by natural selection, but now natural selection is has a harder time telling off the individuals that have the difficult things to get more health care. Have those frequencies risen? Can you tell that from any of the data?
Those are great questions. The first question is whether SNPs are always binary, so are there only 2 versions? And the answer is definitely not. So scientists like to pretend that it's only 2 because computationally it's much easier, and most of the time, it's for most people it's going to be 2, but then there's going to be some small number of people who have a 3rd version or a 4th version because there's only 4 possibilities, and so that is something I think that's being increasingly appreciated as people have done more experiments where they've been able to actually look at every base and seen, in this place that we thought it was only A and T, there are people who also have C. So we generally keep it to a binary when we're talking, big picture, but absolutely. And your second question, which is that basically, if I understood correctly, is natural selection being affected by the fact that we have better healthcare, and so that's going to take things that might normally be selected out of the population and cause them to be selected less. I'm sure there is data on this. I don't know of any specifics. I would say part of it depends on whether what you're talking about is something that, for example, is going to kill you as a child, or is going to for example, PKU deficiency would be one where, so there's a type of disorder where when you're born you can't synthesize a certain molecule that you need, and so if you get diagnosed right away, they can just feed you a diet that doesn't include this thing that's going to be toxic to you. But if they don't know about it, they'll feed it to you, and then you'll die. And so that would be an example of something where maybe there would have been some selection against that if it weren't for medical care. But then again, unless it has any effects until you've had a child, maybe not, because the people who had it would die anyway. So it's a very complicated question, but I'm sure there's a lot of interesting research out there about that. Questions? Great. So we are now going to talk about monogenic obesity. So obesity caused by one gene. And this is going to be in two parts. So first, we're going to talk about how SNPs exert their effects. So what does it actually mean when you have SNP, and why does it cause changes? The second topic is then going to be some examples of monogenic obesity SNPs. So just to remind you, we are here. So we are going to be talking about now rare SNPs that cause a big increase in risk. So first, let's talk about how SNPs exert their effects. So I told you before that DNA was the instructions of life, but I did not tell you what that means. I just told you they were instructions. So I'm going to now explain a little bit what makes DNA instructions, and specifically instructions for assembling proteins. And proteins are the essential building blocks of biology. So you have tons and tons of proteins inside of you that are little workhorses in your body. They carry out a lot of your essential functions. So they're pretty important. So by way of cartoon, I'm now showing you that you have DNA, a little DNA sequence, and that is giving rise to my little green star protein. So the segment of DNA that provides instructions for one protein is called a gene. So by way of illustration, if we look at a stretch of DNA, and let's say this is the sequence that is on this particular stretch of DNA. And we can now say that the sequence in green is gene 1, and the sequence in purple is gene 2. And each of these is going to separately provide the instructions for two different proteins. So the green is going to be for green protein 1, and the purple is going to be for purple protein 2. So you can see that genes are sort of physical segments along the DNA, which are independent and are individually giving rise to these proteins that we need. And to give you a sense of scale, humans have about 20,000 different genes. So there are lots and lots of different proteins in your body that all need different sets of instructions. Okay, so DNA provides the instructions for assembling these proteins. So what actually happens when you change the instructions, right? Because I just told you that SNPs are a single letter change. So that means we've changed the letter in our instructions. So what might be the consequence of doing that? So let's just talk about a few examples of what can happen as a result of SNP. So let's say we have this DNA sequence, which has a C in bolded red. And let's say there are some people who have the sequence below it, which is the same, except that instead of a C, they have an A. So this is a C to A SNP. So one thing that could happen is this. So people who have the C are producing these nice green star proteins. But people who have the A are producing sort of these misshapen proteins where they don't look the same as the ones with the C. So that would be an example of a SNP that's changing the shape of a protein. And for most proteins, their shape is pretty correlates pretty directly with their function. So if you mess up a protein's shape, you're probably also going to mess up its activity or its function. So in this case, we change the shape and activity of a protein. Another example is that you might decrease the amount of a protein being produced. So up top, we had 3 copies of green protein floating around, but the people who had an A instead of a C now only have 1. So pretty straightforward, causing less protein to be produced. And you could probably guess that you might also cause more protein to be produced, right? So first we had 3 green proteins, and now we have 5. And I, as a geneticist, would be remiss if I did not tell you that this would be the last example. So you're probably staring at that wondering what the heck is different in that from the first one, and the answer is nothing. So a lot of SNPs actually cause no changes at all. So just because you see a SNP doesn't actually mean that it's actually doing anything of functional importance. So in summary, SNPs can change the shape and activity of a protein. They can change the amount produced of a protein, or they can cause no change at all. Okay, and that brings me to my next section here, which is some examples of monogenic obesity SNPs. So let's talk about appetite. So as most of you can probably imagine, hunger is critical for survival, right? An animal or a human that doesn't feel hunger is not going to eat, and then they're going to die. So from an evolutionary point of view, you really care about making sure that whatever animal or human you're interested in is going to feel hunger. It's one of the most important sensations that we have. However, if you have hunger, you also need to have an off switch, right? You also need to be able to say, you know, I've eaten enough and I need to now stop eating. You don't just want to have gas pumping all the time, you also need breaks. And so the appetite pathway in humans controls both hunger and then the off switch for hunger. So we're actually going to be focusing on the off switch in the context of monogenic obesity. So what does the appetite pathway in a human actually look like? So when you eat, that causes your fat cells to produce a hormone called leptin. And leptin's role, ultimately, will be to suppress appetite. So leptin is a pretty important hormone. Leptin then travels to the hypothalamus. The hypothalamus is a brain region that controls a lot of primal functions, obviously appetite, but also things like body temperature and sleep regulation. And that's that blue, it's in that yellow circle, it's the blue region, so it's sort of down in the middle of your brain. So leptin is going to travel there, and then it signals to another protein called the leptin receptor. So leptin talks to the leptin receptor, and then leptin receptor passes that signal to yet another protein called MC4R. So MC4R receives the signal from the leptin receptor, and that eventually leads to the sensation of feeling full, which is your body's signal for you to stop eating. Okay, so what do you suppose would happen if you broke the appetite pathway? So if one of those pieces did not function anymore, what do you think would happen? And what happens is this. So this is a mouse that is extremely obese. This mouse arose in 1949 in Jackson Labs, which is they breed mice. So this mouse showed up, and they were like, oh my god, what is going on with this mouse? We need to figure out what's biologically happening here, because this is really, really interesting. So they looked into it, and as some of you may have guessed, it turns out that that mouse had a SNP in the leptin gene. And so that mouse was severely obese because it did not have this hormone that's supposed to start this signaling that's supposed to say, hey, we've eaten and it's time to stop. So as it turns out, this can also happen in humans. So rare SNPs in the leptin gene can cause monogenic obesity. Now this is extremely rare. There are less than 100 known cases of this happening, and where it has popped up, it's usually been in families with intermarriage between related people, like cousins. So this is very unlikely to be something that you would see in your daily life. But it's pretty interesting, and I think also probably helps scientists sort of start to put together this pathway, which I think is pretty cool. And it turns out that this is also true of the leptin receptor, that rare SNPs in the leptin receptor gene can cause monogenic obesity. And finally, also true for MC4R, as you probably guessed, that rare SNPs in the MC4R gene can cause monogenic obesity. And this is actually by far the most common cause of monogenic obesity. So again, this is a very, very sort of wild estimate because these numbers are hard to get correctly. But I would say that something between 2 and 3% of severe and early onset obesity is caused by MC4R, which for a monogenic disorder is actually really, really a lot. So MC4R is a really fascinating example. So you can think of this as if you know that a SNP that causes a monogenic disorder, like monogenic obesity, it's pretty safe to assume that that SNP is really hurting the function of the relevant protein in a really serious way, right? These are going to be SNPs that cause the protein to really just not work anymore. So effectively, if you have a leptin, one of these leptin SNPs, you are removing the leptin part of this pathway. And you can imagine that if you don't have leptin, you are not going to get to the end of the pathway, which is feeling full. And that's the same if you get rid of the leptin receptor or if you get rid of MC4R. So getting rid of any one of these SNPs in the pathway is going to prevent you from the end goal, which is this feeling of satiety, feeling like you've eaten enough. So you can imagine that people who have these SNPs are obese because not only do they have this really strong drive to eat, but when they have eaten, they just don't get the sensation that it's time to stop, and they just continually feel this strong urge for food. And so, in some, as I just established, rare SNPs and multiple genes that are involved in the appetite pathway can each lead to monogenic obesity. So leptin, the leptin receptor, and MC4R. And in these cases, obesity is caused by a strong drive to eat and a lack of feeling full after eating. And I will now take questions again.
If SNPs in leptin receptor, leptin or MC4R can cause obesity, can you also have SNPs that cause like very little leptin to be made, or receptor activity, or very little MC4R that would actually lead to appetite suppression and severe anorexia or something like that?
So the question is, can you have SNPs that cause the opposite effect in these genes, where so you have a loss of appetite as opposed to a gain of appetite? So there is at least one SNP in MC4R that is associated with being protective against obesity. So I don't think it actually causes you to be really underweight or anything, but people who have it are actually less obese than the general population. There have been studies, like the kinds of studies we're about to talk about in the next section, there have been studies on anorexia and things like that, but I don't know if they've found any overlap with these genes.
Is there any research being done for using that chemical as an appetite suppressant in general?
So the question is there any research on using any of these sort of therapeutically? And the answer is yes. So the obvious one would be leptin, to just create synthetic leptin and just put it into people. That does not work. And the reason it doesn't work is because most obese people actually develop leptin resistance. So they actually have very high levels of leptin, but their cells just don't respond to it anymore. So, which is too bad, because researchers thought they found the holy grail when they found this, and then it turns out it works in people who have a SNP in the leptin gene. So you can treat those people with artificial leptin, but pretty much nobody else. There is actually there's therapeutic development going on now by a company called Rhythm on MC4R and trying to make that receptor functional. And I think they've mostly been looking at it in the context of people who have one of these monogenic obesity disorders and not garden variety polygenic obesity, but I think that there have been some pretty promising results there in clinical trials. All right, so we can now have intermission, so you are free of me.
For now.
For now. I actually
Yeah.
So yeah, I think but generally they're all not.
Oh, yeah. There's one of this.
Yeah, but I think
I think it was way more.
and then people can question if you've never been.
Yeah.
Oh, well, the options you
No, I Okay. Yeah. 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 No. Yeah.
It makes sense that way. Right, like it seems, but.
Oh, right, yeah, no, this one.
So that should happen.
You're all set.
I think that's how we all but I heard. I mean, and maybe people know where it goes. People ask for it.
They're right.
That's why I know.
I know, right? I'm just gonna stop my face with chips. Let's talk about
So you don't have data on this, let's say, but that's like a pretty obvious one. Because where is this muscle factor?
No, it's my fault. I like had a chaotic morning where like in my head my class started at noon, but then I realized at like 10:30 that it actually started at 4:30. So I was gonna like have a dinner and I couldn't. But next week.
It's such a pain to put on camera.
So where does it normally go? It's just normally here. We can see that all happen.
Yeah, well, I mean, that would be. Well, you need it not.
And do you try to fit it into it?
So usually only one.
And it's gonna save that. I'm gonna check her. It's funny. It's gonna drink shot in there. Is it used for anything else?
Oh, sorry, yeah, the reason there's.
Got it.
Yeah, no, it's a demand issue.
Sucks.
We try to overweight.
Yeah. Who else?
Any other student.
Let's take them all out.
Down, I think.
Sure.
Yes. I get emails from them. Is that it's I don't even really know how that's different from like DMS.
So DMS.
Okay, we're also
Right. So the pills includes DMS and also.
so fully.
It also has like a lot of flavors. Which is why for organic life sciences. So it also has like.
Okay. I wish there was like a Venn diagram. So I think they explain all of that to us. Well, and they explain to the orientation like day one.
Are you ready for next minute?
Are you ready?
I'm ready.
I'd like you to know.
It's,
Yeah, get out.
Okay, just as we're getting ready to start up again, I'll just remind you that we do have handouts, flyers, and surveys. Oh, we're on that table over there. So if you do get a chance, grab one. Feel free to get up and grab one now. We're still finishing up. But we would love it if you would leave us feedback. You could also come up to me in person, but I forget things easily, so. Thanks.
All right, welcome back. So in this last section of the talk, I'm going to be discussing polygenic obesity, which you'll remember is obesity caused by multiple genes. And I'm going to, again, divide this into two parts. So first, I'm going to talk about genome-wide association studies, which are the method that we use to look at polygenic traits generally. And then I'm going to talk about some findings from a large genome-wide association study of BMI. Okay, so just to remind you, we are here. So we're talking about common SNPs that cause a small increase in risk. So most of the time, as I've discussed with you, obesity is caused by more than one gene, plus diet and exercise and other lifestyle factors. So how can we figure out what those genes are? So the key insight here is that because we're looking for tiny effects, so SNPs with a very, very small effect on obesity risk, we need lots and lots of people to find them. And I'm going to illustrate that to you by way of an extended coin analogy, which is everyone's favorite thing. So if I flip a coin 100 times, and I get 80 heads and 20 tails, and I asked you whether you thought that coin was biased, you would probably say, yeah, because you got heads 80% of the time, right? And in an unbiased coin, in a fair coin, you would expect to get heads 50% of the time. And that's such a big difference that you would say, I'm not exactly confident of how biased this coin is, but it's definitely biased, right? It's not fair. So you would say, that's probably biased. If you flipped a coin 100 times, and you got 51 heads and 49 tails, and I asked you whether you thought that coin was biased, you would say, well, I got heads 51% of the time, but it was really I got 2 heads more than I got tails, right? So that seems like that could be well within random chance for a coin that actually was fair, right? So you'd probably say, you know, this doesn't seem biased. I suppose it's possible that there's a small amount of bias, but with the number of flips that I've done, I'm really not confident that there's a meaningful difference here. So that's uncertain. But if you flipped a coin 1 million times, and you got 510,000 heads and 490,000 tails, and I asked you if that coin was biased, you'd say, okay, well, I got heads 51% of the time, which is the same percentage of time that I got heads for the last coin. But this time, I have a difference of 20,000, right? I got heads 20,000 more times than I got tails. And so even though this is only a 51% chance of heads, in terms of just the math of it, and that's only 1% above what I would expect from a 50-50 shot, I've now done this so many times, and it has come out so consistently as being just a little bit more heads, that I'm actually going to say that I'm pretty sure this coin is biased. It's not biased by much. It's a very, very small bias, but the bias is there. So this is probably biased. So looking for SNPs with very, very small effects is like looking for a coin that gives you heads 51% of the time, because it's such a small difference relative to what you would expect from random chance that you need to test it in as many people as you possibly can to be sure that what you're seeing is real and isn't just statistical chance, right? Because the more times you do it, the more confident that you can be that some small change is meaningful and not just statistical fluctuation. So to sum that up, if we made this graph and we put the size of the SNP effect along the x-axis, so from a small effect to a large effect, and how many people needed to discover that effect on the y-axis, it would look something like this. So the smaller the effect you're looking for, the more people needed in your GWAS, Genomewide Association Study, or to talk about it, to discover it. And conversely, of course, if you have a large effect, you don't need that many people to be sure that what you had was real, because the difference is going to be so big that you don't need to test it that many times to be sufficiently confident. Okay, so that brings me to Genomewide Association Studies, which we abbreviate as GWAS. So this is a method that was first developed in about 2005, so not that long ago. And the principle is that we're going to test thousands of common SNPs across the genome with association to a trait, and in this case, that trait is BMI. So Genomewide Association Study, which is sort of a terrifying sounding term, is actually exactly what it sounds like. So you're looking across the genome, so genome-wide, for association. And so the idea is that you're literally going to go SNP by SNP and say, does this SNP appear to be associated with an increase or a decrease in BMI? So how do we get the data for GWAS? I had another slide. So GWAS is a fairly, really sort of an incredibly explosive thing for something that's only been around for 10 years. So it's the basis of 2,794 publications as of a couple days ago. And it's found 32,320 unique SNP trait associations. And that's for hundreds of different traits. So height, cholesterol levels, cancer, age of menopause, anything you can think of. If someone can get their hands on data for enough people, someone has done the GWAS of it. And this is really what we mean when we say the genomics revolution, right? This is looking at really big data. This is looking at thousands and tens of thousands and hundreds of thousands of people. So this is a good moment to remind you that everyone has 2 copies of every SNP. So you have 1 copy from your mother, 1 copy from your father. So with a T to C SNP or T to G SNP example, you could be TT, you could be TG, or you could be GG. Those are the 3 possible options. So in order to perform a genome-wide association study, we need to obviously collect some genetic data from the people who are participating. So we collect either blood or saliva, and then we get the DNA from that sample, and we then test thousands of common SNPs to determine what version this particular person has. So this image that I'm showing you is a little cartoon that I drew myself, which is, we call this a microarray, and I don't have time to get into the details of how this works. But essentially, every spot is testing a different SNP. And the color of that spot is going to tell you whether for that spot, for that SNP, the person has, for example, TT, or TC, or CC, because there are only 3 possible versions, so there are 3 different colors. So for example, if we looked at the yellow dot there, so let's say that's some C to T SNP somewhere in the genome. And the color is going to be red if this person is CC, or green if they're TT, or yellow if they're CT. So this is yellow, so that means that for this particular SNP, that person is CT. So you do this across all of your people, all of your participants, and then you're ready for analysis. So let's say we have this SNP, which I'm going to call SNP1, and this is a T to A SNP. So we have the T in purple and the A in red. And we're now going to make a graph where along the x-axis is the versions of a SNP a person might have. So there are 3 possibilities, right? TT, TA, or AA, depending on what you inherited from your parents. And along the y-axis, we're going to have BMI. So we have a measurement of the BMI of each person. So we then can plot the relationship between the person's versions of the SNP and their BMI. So each dot represents 1 person, and the person is plotted against which versions of the SNP they had against their genotype, or sorry, against their BMI. So you can see, so you would look at this and you'd say, all right, does having an A instead of a T increase your BMI? And in order to answer that, you would sort of say, does it look like there's a relationship between the x-axis and the y-axis on this graph? And you would draw this line. So you would say, it does kind of look, right? Like if you have a TA, you have a higher BMI than if you have a TT. And if you have an AA, you also kind of look like you have a higher BMI than if you have a TA. So it does kind of seem like having an A instead of a T in this particular spot increases your BMI. Now you can also imagine that if we looked at a different SNP, so this is SNP2, C to G. So if we did the same thing, so we have our 3 possible versions of the SNPs, along the x-axis and BMI along the y-axis. And we look to see, is there a relationship between which SNPs you have or which versions of the SNP you have and your BMI. And you would draw this line, which is pretty flat and boring. So if you asked, does having a C instead of a G increase your BMI, you would say no, right? It looks pretty random. It doesn't really look like there's a relationship between which version of the SNP you have and what your BMI is. So we do this for every SNP that we have information for in the genome. And we use statistics to sort of quantify how confident we are for each SNP that this SNP is related to BMI. So that takes me to the last part of this talk, which is talking about some actual findings from a genome-wide association study of BMI. So in 2015, a GWAS of BMI containing 340,000 people was published. I just want to emphasize that that is a totally insane number of people. That is one of the biggest GWASs that I'm aware of. And they found 97 groups of SNPs associated with obesity. So just to sort of remind you what that means, the more obesity SNPs you have, the higher your risk of obesity is. So that's 97 spots where you might have a very small increased risk of obesity. But I should say that these SNPs only account for about 2.7% of the variability in BMI between individuals. So this is really not accounting for big changes, so just keep that in mind. So let's talk about a specific finding, and then let's talk about a general finding. So let's start with the specific. So one of the things that they found was that there are, it turned out, were common and small effect variants in MC4R. And you will remember that we already talked about the fact that there are rare and large effect variants in MC4R that can cause monogenic obesity. So that's pretty cool, right? Because that means that the same gene is coming up in sort of two different ways as being relevant for causing obesity. So just to sort of compare this for you, so from GWAS, these polygenic SNPs that we're finding, we find common SNPs in MC4R, whereas the ones from these monogenic studies are rare. And the common SNPs that were found by GWAS have small effects, so a tiny increase in obesity risk. Whereas the rare SNPs that cause monogenic obesity, by definition, cause large effects. So a massive increase in obesity risk. And finally, we can talk about our general findings. So one of the nice things about a GWAS is that you get, if you're lucky, you get a lot of potential associations with the trait that you're looking for, right? You don't just get you're not just looking at one gene, you're looking at all of the genes. So you have a lot more material to work with in terms of cool things that you might be able to test about this trait that you're looking at. And so there are a lot of really cool and interesting questions that you can ask from having this kind of data. But one of the most basic is, of all of your potentially associated genes to this trait of interest, so for BMI, what functions, what biological functions do these genes share? And the reason that we care about this is because you can imagine that if we know that a SNP in a gene is likely to be associated with causing something like obesity, and we have a bunch of those, then it probably also means that those genes are interesting for the purposes of wanting to know what causes obesity, right? Because we know we have evidence that suggests that this gene might be causing obesity. So if we want to know what's causing obesity, we should figure out what these genes are actually doing, right? And from a therapeutic point of view, you can imagine that's also very important. That we want to know what do these genes have in common, because that's going to point us to the right biological processes. So when they asked this question about these 97 groups of SNPs, they found something really unexpected, which was that there were lots of genes that affect the brain. And specifically, they found genes that were involved in a process called neural signaling. So what is neural signaling? So if you are on your cell phone and you want to talk to your friend, you will call their cell phone. And your phones are going to send communication through a signal, right? That's what's happening between these phones that's allowing you to communicate with each other. So in the same way, if we look in the brain and we look at a neuron, which is just a brain cell, that neuron is going to need to talk to other neurons. That's a really, really fundamental and important part of neurobiology, is that your neurons all have to be talking to each other. So this neuron is going to send a signal, just like your cell phone sends a signal to another cell phone, to another neuron in the brain. And this process is happening all the time throughout your brain. This is one of the things that keeps you alive. It's very important. So, okay, so if the genes relevant to this process have SNPs in them that seem to potentially cause changes in BMI, what effect do we think that might have on this process? So if you change a gene that's involved in neural signaling, you might do something like change the strength between neurons, right? So how strongly do these neurons talk to each other? How often do they talk to each other? Or you might change the patterns of communications between neurons. So which groups of neurons are talking to which other groups of neurons? So it seems, for reasons that we actually still really don't understand, that these processes are somehow relevant to causing obesity, and that's actually really something that we're actively working on trying to figure out right now. So in conclusion, GWAS has discovered many small effect SNPs that affect BMI. MC4R has both small and large effect SNPs within it, and many of the genes discovered by GWAS for BMI affect brain signaling. But to take a step back for a second, there are some core principles that I hope you can all take away from this. So first, some core principles about obesity. So as we've talked about, obesity comes in a polygenic and a rare monogenic form. Monogenic obesity comes mainly from the appetite pathway, so these genes that have strong roles in appetite. They were able to find through GWAS 97 groups of SNPs associated with polygenic obesity. And the more obesity SNPs you have, the higher your risk is of obesity. But of course, your lifestyle matters a lot too. But even bigger core principles, kind of about genetics as a whole, once again, there is a very strong brain component of obesity, which people were really not anticipating, right? So there's the appetite pathway, the hypothalamus that we talked about, and we kind of already knew that, but this neural signaling thing was totally unexpected to most of us, and really suggests that while we think of obesity as really being something maybe about metabolism or digestion, there's a really, really strong brain component, which I think is pretty surprising and interesting. So, and in general, when we think about polygenic traits, because they involve small effects, we really need GWAS to study polygenic traits. You really can't get at these tiny effects without an approach like GWAS, where you use all these people. And so that really illustrates both the power of big data to move science forward, because in these 10 years of GWAS, we've already learned about hundreds of different traits, but it also illustrates that it's necessary. Because in this area, at least, we can't make progress without these types of approaches because of the nature of what we've discovered, which is that these things all have these really small effects. And as samples grow larger, our ability to find relevant genes and create, hopefully, new medicines will only improve. And the next BMI GWAS, which my boss is one of the people in charge of conducting, will hopefully have over 1 million people in it. So if you can even imagine data on that scale, because I can't. So the future is bright, and I hope that you've all learned something interesting today, and I'm happy to take questions again.
Going back to the beginning, you're talking about body mass index. Why is it weight, proportionality weight height squared, not weight? You'd think that if somebody's taller, you have like slightly larger bone structures, it would be a good that.
So the question was, why is BMI calculated as weight divided by height squared, not weight divided by height cubed? And the way that BMI was developed was empirical. Like I think they were looking for a measure of weight that was relatively independent of height, and it turned out that squaring it gave them the best relationship. But it's one of these things also where it works the best for people in the middle. Like if you have sort of an average height and average weight, and if you're really short or really tall, it doesn't work as well. So it's not. I mean, it's not a perfect measure.
I mean, I'm saying somebody that's like 7 feet tall, if they're also a little wider.
Yeah, I think if you're
like 5 feet tall.
If you're 7 feet tall, BMI is probably not going to be the most accurate measure. But I don't even think it would. I mean, even if you use cubed at that point, I mean, you're so far outside. Yeah, you may need to. You would probably need to sort of make a different measure for people who are of sort of the extremes of the height distribution. But that's a fair point. Yes.
So you mentioned that at the beginning that certain drugs can affect weight gain loss. Are any of those known to modulate the neural signaling that was identified through that GWAS?
So the question was, are drugs that are known to modulate weight gain, were any of the sort of the targets of those drugs found by GWAS? So for drugs that actually, it's actually a really interesting story there. So there's a drug that is pretty effective for obesity called topiramate. Which is pretty new, and topiramate was originally an anti-epilepsy drug, so it has really, really strong roles in brain signaling, obviously, because epilepsy is all about, crazy electrical signaling in the brain that can't be stopped in the right way. And so people didn't know, and still don't know, why topiramate works, because it just seemed like such a weird thing that something that would work in the brain in that way would affect weight. But then this GWAS came out. Maybe there is actually like a real relationship between this brain signaling that this drug is affecting and BMI. So, and the other thing is that other drugs that affect obesity, a lot of them are actually affecting like norepinephrine and serotonin, which are neurotransmitters. So I think that the drugs might have already kind of given you a hint that something was going on in the brain.
I know some people have been using antidepressants. A lot of them have had these severe weight gain and cholesterol.
Yes, the comment was just that she knows a lot of people who are on antidepressants, and one of the big side effects is weight gain, which is totally true. It makes sense because it's affecting these same systems that I was just mentioning, like serotonin and possibly norepinephrine. Thank you.
But actually, I mean, I'm sure like when you were preparing for this, you have to I know this. No, I know. That's really ironic. That's funny. I started the French fries, like for very long. I was like, I'm starving. I was like not being.
Oh my god.
So whatever works best. I'll have to remember that. Yeah, it's like when I go to like diet, and then when I start talking about different diets. I always said we should talk about it. Hi, nice to meet you. Are you a med student or okay?
Sorry?
So it's not just control, it's